So welcome from Germany or Korea. Well, no, it's just a bunch of flags behind me. But welcome to the AP Soil Compaction Lab. So what is soil compaction? Obviously, making something more compact, you're compressing it, right? The idea with soil compaction is that you now, as you've compressed the soil, it could be with any kind of soil, by the way, that now water infiltration through that soil is going to be lessened. It's not going to be able to flow through the pores as well as it normally would if it's not as compact. So water retention will increase, water infiltration will lessen. Speaking of lessening, what is the lesson today? Well, okay. We're going to have a lab that really goes into a student-generated experiment that deals with soil compaction. And there are going to be different kinds of soils that are available. And what does really a different kind of soil even mean? Clay, for example, if you have it available, which is not always so easy to find, and I highly advise you not against using those clay pellets. That's very different. But clay is very fine and is famous for soil compaction because it also has some cohesion where it sticks together. And although the grains can move past each other, maybe even a little bit better than sand, from my understanding, it does stick really well together and it's really small. So what happens when it gets really waterlogged is it sticks even better together. Think about charges and things and how that works. And if it all sticks together, obviously, then you don't get infiltration. You get a lot of water retention, which again is what clay is famous for. In this lab, they also have sand as a potential grain size. That's what you really should think about these as. These soil types are based upon grain size, and sand is much larger, and the water just runs right through it. So although pore size is very much more complicated than this, we're not really talking about that in such specifics right now. We're just talking about soil compaction. So sand is often thought of as harder to compact for a type of soil. And then, you could have loam, and it really depends. I even bring marble chips into this. Depends on what you want the students to work with or what the students want to work with. Talking to teachers and students, whoever wants to think about this way. But if you are a student, depends what you want to work with and what your teacher has available. If you're a teacher, depends on how you might want to tailor this experiment. So this lab actually does, in a way, very much so, deal with pore size and porosity, which are different. Pores are the holes in the soil, and it's just to draw. That doesn't work. Yay. I was just to draw here. What you'll see is that's water running down between the different grains of soil. And if I have large grains, I might well have large pores. If I have smaller grains, and oftentimes these are not isolated, like clay is smaller, there'll be smaller pores for the water to run through, but there'll often be more of them, which means it will have more porosity overall. We also differentiate between smaller pores called micropores and larger pores called macropores. And with soil compaction, we're really getting rid of those macropores. Those pores are necessary for air and water infiltration, and that's what we're really looking at in this lab. So one skill that really is emphasized in AP Environmental Science is being able to design and conduct your own experiments. And really that experimental design is tested in FRQs and multiple choice. So you have to be able to do this. And what this lab does is it really tests the student's ability to design their own experiment, to test soil compaction levels based upon soil types. It tells you to use something like chickpeas, and that's what I use, because the radical is easier to see and measure because it's a fairly big seed type and it needs to be pre-germinated by the teacher. I did it, yeah. But that takes 48 hours in advance. And then there might not be a radicle showing, but it should be ready to germinate when it's planted. And some of the time you do get some radicals, just a little bit. But they need to be measurable. You could even look at root hairs. And the students are really the ones coming up with their design. They need to be aware of and understand what their independent variable would be, dependent variable, and what they need to keep constant. So they need to understand their variables and what to keep constant. And that's, when we talk about details of designing an experiment, that's really important actually. How many things need to be kept constant to make it a really valid experiment. We have access to a greenhouse here, which is nice, but this can be done without a greenhouse quite easily. It's better for keeping conditions stable and constant, but again, if they're all in the same conditions, it kind of works out anyway. And these seeds grow pretty well, so you just need a nice space where you can set this up, or really where the students can set this up. This is really student-centered. So a couple of last minute things about the lab I want to talk about quickly. One, just remember it is an experiment that the kids are supposed to be designing and conducting with certain parameters in place. But that doesn't mean you shouldn't guide them. And if you're watching this as a student, same idea. You're supposed to be doing this on your own. Listen to the guidance of your teacher. Two, I would not recommend changing the hypothesis. Think that you shouldn't at this point, and stick with the specifics and the hypothesis that were laid out beforehand. So if there are going to be stems, sometimes there are. A lot of times there are. That's okay. You could think about measuring stems, or you could go stick with the radical, which you really should. So I think that's really a good move.